Move on. So to put it kind of in a vulgar way, racial passing is the idea that one would fraudulently portray an identity that one does not have kind of an authentic claim to. So for example, a black person who might be passing for white, or a woman who may be passing for a man. It's gender passing. And we can talk about this a little bit later if you want to, but I'm focusing particularly on racial passing, and particularly on racial passing as it comes from a narrative that is embedded within U.S. chattel slavery and anti-black racism. So this presentation is part of a dissertation chapter on race and gender passing in the U.S. Narratives of passing have often been examined through the dynamic of individual strategy in favor of social mobility, access to love denied, or identity politics. However, my dissertation argues that the passing narrative is much more complex, rarely confined to an individual's autonomous will, and far more entrenched in state building and concepts of social progress than previously imagined. To begin to trace the workings of this particular modality of race and gender, one must shift focus away from the actions of the passing individual and towards the accusations of passing itself as a technology of disciplining and surveilling bodies and regulating populations for the good of the nation. In that vein, let's move away from the desire of the passing subject for a moment and back to our own. What is key in our attempts to see, classify, and regulate our bodies is the sort of anxiety, excitement, and panic that undergirds these practices. This paper asks us to consider the moral economies in which science traffics that embed our desire to know and our desire for mastery in histories of black female sexuality and labor. To do this, I want to direct our attention to the story of the HeLa cell contamination and have us read the moment as an accusation of passing. This is one in which the passing offender is not a subject but an object, basic biological material. As many of you probably know by now, Henrietta Lacks is a black woman who died of cervical cancer in 1951 in Baltimore. Without her consent, Lacks' doctor gave a biopsy sample of her cervical cells to George Guy, a cell tissue researcher at Johns Hopkins, for the use in laboratory studies. Because they were able to reproduce on their own in the laboratory, something no other human cells were known to have done before, Lacks cells were traded globally and became the basis for numerous medical advances, including the polio vaccine, genetic cloning and mapping, and in vitro fertilization. The story of Henrietta Lacks and the HeLa cell line is not a new one. Upon the discovery of the cell's regenerative capacities, narratives of the cell line and Lacks' identity emerged fairly soon and quite strategically. In fact, one of the earliest narratives was publicized specifically in order to garner support and funding for the Foundation for Infantile Paralysis and its efforts to lead the initiative to find a vaccine for the polio virus. Roland Berg, the foundation's director, convinced Guy that the story of the HeLa cells must also include Henrietta Lacks. How else would the foundation garner the needed interest if the cell line discovery story were not also a personal interest story? During this early period, however, blackness was not central to the narratives about the HeLa cells. In fact, the identity of the woman from whom the cells originated was not all that clear and often misstated. Some authors referred to her as Henrietta Lakes or Helen Lane. Others named her Helen L. Further, it seems that Lacks was assumed by many to be a white woman, as the foundation no doubt tacitly promoted her, the quote, unsung heroine of medicine. In the science literature, HeLa began to refer not to the specific cancerous cells of a black woman's cervix, but to the universal or, quote, generalized human or cellular subject. With this narrative, any specific historical or cultural meaning attached to the positionality of lax as a black woman and a domestic and agricultural worker is emptied out. In this period, HeLa's was a story of how any woman, presumably of course a white woman, could make a sacrifice for scientific advancement and thus for the progress of the nation. This raises the question, how then did the HeLa cell line become fixed as black? Rebecca Skloot's now widely read book, The Immortal Life of Henrietta Lacks, has a chapter on this particular moment, and yet she does not elaborate on its cultural or political significance. She does, however, recognize that it was violently important for the scientific community, and thus named the 5-page chapter she dedicates to it, the HeLa Bomb. This is also the phrase that the cell culturist community used at the time to refer to this moment. As the story she tells goes, in early 1966, at the Second Decennial Review Conference on Cell Tissue and Organ Culture, Stanley Gartler provided some upsetting news to the conference attendees. In his presentation, he revealed that the HeLa cell's capacity to pass themselves off as other cells and contaminate the samples with which they come into contact. Thus, results of the studies scientists had drawn while working with what they thought were various other cell lines had to be called into question, and years of work would need to be completely disregarded. Gartler recommended that attendees return to their labs and review their cell and tissue cultures for HeLa passing and contamination. Race was at the center of Gartler's investigations. He compared both phenotypes and genotypes of 20 sample cell lines and found them to be sharing components of the same phenotype. Further, the phenotypes comprised variants that had been determined to appear most frequently in the, quote, American Negro male population. It was through the framework of racial biostatistics that Gartler concluded that the sample cell lines had been taken over by HeLa cells and marked by a phenotype most commonly held by black men. He made no comment about sex discrepancy. Instead, the narrative told in the scientific community and beyond was one of white cells in danger of contamination and disappearance by HeLa cells, which are black. The fallout from this accusation was both personal and professional for the cell tissue community. As Robert Stevenson, who later became president of the American Type Culture Collection, put it, Stanley Gartler, quote, showed up at the conference with no background or anything else in cell culture and proceeded to drop a turd in the punch bowl. This in itself is a contaminating act that introduces the abject into the space of scientific sterility. One of the affected cell lines that Gartler identified was fellow scientist Leonardi Flicks' wish line. These particular cell tissues originated from the amniotic sac of one of Hayflick's daughters. Hayflick's concern for his cell line turned to racial paranoia. He called his wife during a conference break to ask whether he was, in fact, his daughter's biological father. As he retold the anecdote during his own presentation, quote, she assured me that my worst fears were unfounded. The room reportedly, quote, erupted into laughter, and no one said anything else publicly about Gartler's findings, end quote. Racial and sexual anxiety turned to comedy as the threat of miscegenation was covered over. It was not just that HeLa appeared to the scientific community as a passing actant, but that it had the capacity to make passing subjects out of others. Hayflick's daughter, for example. The link between HeLa contamination and the destruction of professional careers and scientific progress emerges through the racialization, gendering, and hypersexualization of the cells. For instance, one journalist wrote that a letter from Nelson Rees, a biologist who dedicated his work to the detection of HeLa contamination, was like, quote, a note from the school nurse informing the parents that little Darlene had VD, end quote. Thus, boundaries between Henrietta Lacks' blackness and femaleness and the HeLa cells were continuously blurred. One gathers that Nelson Reed's dedication to unveiling HeLa was a toilsome task, not simply because it required him to be the bearer of bad news for otherwise good scientists, but because of the racial, gender, and sexual anxiety that the HeLa Bomb produced in the cell culture community. He acknowledged to Michael Rogers, quote, "I hoped I'd never have to look another HeLa in the face." So why has the uncovering of this cell line come into the discourse through the metaphor of a bomb? And what biopolitical work does this metaphor do? To take the bellicose reference literally means to assume that HeLa passing is a warlike moment in the midst of compiling of biomaterials for medical research. Foucault argues that medicine itself is, quote, a political intervention technique with specific power effects. The underlying biopolitics of defending society is a matter of, quote, destroying that sort of biological threat that those people over there represent to our race. Thus, to think of HeLa passing as a bomb within the historical context of the time, 1966, is to think the passing threat together with other bomb threats and detonations of the time. The Cuban Missile Crisis in 1962, the escalation of the Vietnam War between 1963 and 1969, and the bomb that ripped through the 16th Street Church in Birmingham in 1963. During the period of the Cold War and Black uprisings, the threat of a nuclear bomb gave us the practice of duck and cover, along with the hypervigilance against the communists that might be lurking in our own backyard, quote, red under the bed. Can the threat of a black female worker who passes undetected be projected onto these simple cervical cells? Within the narrative of passing, HeLa represents a danger for the sort of categorization necessary for biopolitics. Within this context of war, the threat of the HeLa passing is the possibility of not knowing who the other is, not knowing how to identify and target the other, or to discover that one is the other. The acts of the passing HeLa cells constitute a circuit of non-human insurgency, allegedly an act of sabotage on the very construct that enables biopolitics. Surveillance and regulation then become critical techniques that bind the practices of medicine and science to a larger national and geopolitical project, one that biologists see themselves as taking on. For example, Roland Nardone, a cell biologist at the Catholic University of America, is referred to in one article as, quote, the Paul Revere of cell contamination. In other literature, HeLa is described as, quote, slipping behind the Iron Curtain and contaminating cell cultures in Soviet labs. Nelson Reed's publication of HeLa passing into the Soviet Union was believed to put Cold War relations between the two powers in further danger. To imply that Soviet cell culture samples were really those of a black American woman was considered, quote, a gratuitous attack on the Russians. For his work in detecting the HeLa fallout more broadly, Nelson Reed is described as embarking on a, quote, lonely crusade to trace HeLa contaminants. The reference to a crusade is particularly telling in this Orientalist narrative, as it sutures the discourse of the biomaterial of the black woman's cervix to signifiers of another radically different other who is neither Christian nor white. While the HeLa contamination emerges as a crisis of insecurity, the passing narrative attempts to contain the unwieldiness of blackness and particularly black female sexuality. The unveiling is actually part of the modern colonial and scientific project of surveillance, once again reasserting the human's ability to enact the nature's order of things. Thus, the HeLa passing accusation is a story to reassert the predictability of non-cancerous human cells. Gartler's presentation in 1966 concludes with just this argument. He reasserts that his findings demonstrate, quote, the remarkable stability of normal human cultures, That is, the virtual absence of spontaneous cellular transformation among them. He suggested, quote, the incorporation of stable genetic markers in material to be cultured is the best guarantee against contamination. What scientists needed to do was identify and categorize the stable cell characteristics that would also allow them to grasp the truth of the cell and limit black female hypersexuality. Once this was accomplished, there would be no need for cases of, quote, mistaken identity or, quote, identity theft, as Chatterjee would later describe the HeLa phenomenon. Once this story is secured, HeLa can again be put to work to serve the scientific and imperial project. Got one quick page left. And so this presentation might be thought of as an invitation to consider cellular biology and genomics as already not only participating in broader philosophical arguments around the universal and the particular, but producing ways in which we manage nature culture and its relationship to the state and geopolitics. Can we understand the modality of passing as one that is embedded within modern scientific inquiry? Is it through this narrative that new scientific citizen subjects emerge? And is it through this narrative that science must both disavow race and hold on to it for dear life? We must also ask the question, what is the fallout of the HeLa bomb within spaces of knowledge production? HeLa occupies the position of the universal human subject, a white woman. Gartler's statistical analysis of the cells briefly rendered them a Negro man. Then science and science journalism described the cells as hypersexual black women. The pathways of toxicity, as Chen would describe them, or the fallout of the HeLa bomb, thus, have an effect that animates the cells as potentially fathering a young white girl, Flicks' daughter. Clearly, the cells are not, quote, containable within a given trajectory of movement and desire. If, as Flick feared, HeLa is both a passing actant, possesses the ability to create passing subjects out of others, and if HeLa also has the capacity to destroy, to quickly lead its host's body to a cancerous death, how might we understand the political force of these mobilized cells? Of course, one must reject imbuing HeLa with an anthropomorphized liberal agency. At the same time, following the lead of Mel Chen and other new materialists, one might ask about the ways in which the HeLa cells have been animated. That is, what is the agency of HeLa, a cell line that is queerly passed through race and genders and contaminates along the way?